Hello, my name is Joshua Mgavi. I welcome you to Reproduction in Living Organisms. Question 36 to 40. Discuss the negative and the positive feedback mechanism. Regulate the ovarian and the menstrual cycle and explain how the cycles are affected if implantation of a fertilized egg occurs. B. Describe the events that take place in reproduction, beginning with the production of gametes, ending with the successful implantation of the blastocyst. Discuss the negative and the positive feedback mechanism. Regulate the ovarian and menstrual cycles. Explain how the cycles are affected if implantation of a fertilized egg occurs. The low levels of estrogen signals the beginning of the menstrual cycle by triggering the negative feedback response. The hypothalamus initiates the response of secreting gonadotropin-releasing hormone. Gonadotropin-releasing hormone, in turn, stimulates the anterior pituitary to release the follicle-stimulating hormone, FSH, and the luteinizing hormone, LH. As part of the ovarian cycle, the follicle-stimulating hormone stimulates the development of the egg within the follicle, completing meiosis 1, and also stimulate the follicle to produce estrogen. In a positive feedback reaction, the rising of estrogen levels, the anterior pituitary secretes additional luteinizing hormone after stimulation by the hypothalamus. This spike of luteinizing hormone in the middle of the cycle causes ovulation, the release of the egg, actually the secondary oocyte of the follicle. After ovulation, the follicle, now called corpus luteum, continues to produce estrogen and progesterone. These hormones regulate the menstrual cycle by stimulating the thickening of the endometrium, that is the inner lining of the uterus. In response to continued high level of progesterone and a negative feedback response causes the anterior pituitary to stop the production of luteinizing and follicle-stimulating hormones. As a result, the follicle stops producing estrogen and progesterone, and the endometrium is sloughed off. If the egg becomes fertilized and implants in the endometrium cells, the developing embryo secretes human chorionic gonadotrophin, which induces the corpus luteum to continue the production of progesterone, which in turn maintains the endometrium. Part B. Describe the events that take place in reproduction, beginning with the production of gametes, ending with a successful implantation of the blastocyst. Gametogenesis. Diploid spermatogonia grows rapidly to form the primary spermatocytes, which undergo the first meiotic division to form the secondary spermatocytes, which are still diploid. Then the second meiotic division to form the haploid spermatids, which then differentiate into sperms. In the female, the diploid oogonia grows into a primary oocyte, then reaches the prophase of the first meiotic division prior to birth. The ovaries thus contain primary oocytes, which are haploid, and these only undergo second meiotic division after fertilization by the sperm. Mating. Appropriate courtship behavior synchronizes sexual activities between the female and the male. The sperms are produced from the testes, travel through the epididymis, viability maintained by the secretions from the prostate and the Cowper's glands, released through the urethra into the female's vagina during ejaculation, travel by peristalsis up to the female genital tract into the oviduct. Fertilization. Acrosome of the sperm penetrates the vitelline membrane, leading to the formation of fertilization membrane and combination of two haploid gametes to form diploid zygote. Development of the zygote. The serial movement of the zygote down into the uterus. Within 1 week, holoblastic cleavage to give the blastomeres and the blastocyst. Implantation. In the uterine lining, development of the outer layer of the blastocyst into trophoblastic villi, which project into the uterine lining, is formation of the amnion, chorion, and allantois, the last two combining to form the placenta. The conceptus is maintained viable by a well-balanced hormonal control. 37. Describe the mechanisms in flowers which favor inbreeding, outbreeding. Part B. State the genetic consequences of inbreeding and outbreeding in flowering plants. Part A. Describe the mechanism in flowers which favors inbreeding. 1. Movement of stamen or the stigma when the flower gets older, so that the stigmatic surface touches the anthers or some part of the flower coated in pollen. Cleistogamy. The flowers do not release the pollen onto the ripe stigmatic surface before the flower buds open, thus favoring self-fertilization. Floral structure. Some flowers are arranged that their corolla tube separate from the plant. The anthers rub against the stigma, transferring pollen. Describe the mechanism in flowers that favor outbreeding. Dichogamy. This is where the stamen ripens before the stigma. The ovule, that is called protandry, or the pistil ripen first before the stamen, which we refer to as protagony. Unisexual flowers, dioecious flower plants, which limits inbreeding. Floral shape. Some highly specialized flowers, the stigma is exposed to allow insect entry. Incompatibility or self-sterility. Conspicuous petals and sepals. Nice smelling to attractive scent production. Part B. State the genetic consequences of inbreeding and outbreeding in flowering plants. Let's look at inbreeding. Reduces variation as a result of buildup of homozygotes. There is less evolutionary potential due to the stabilizing selection pressure and less adaptability to the changing environment. Species are more vulnerable to diseases due to homozygosity. Possible buildup of harmful recessive genes and their expression due to greater chances of recessive offsprings. State the genetic consequences of inbreeding and outbreeding in flowering plants. Want to look at outbreeding. There is less vulnerability to diseases for high colonization and survival chances. Greater evolutionary potential due to increased genetic variation. Greater adaptation to the ever-changing environment and increasing chances of survival. Reduced chances of harmful recessive genes expressing themselves, hence hybrid vigor. (38) Distinguish between genetic diversity and species richness. Roman B, Part 1. Outline the various ways used in maintaining genetic diversity via breeding programs. Part 2, Roman B. Explain how breeding programs and reintroduction schemes will help to increase the population in the wild. Part C. Outline how the acrosome reaction is triggered in human reproduction. Part 2. Polyspermy is prevented after acrosome reaction. Let us start with distinguishing between genetic diversity and species richness. Genetic diversity considers one species, whereas species richness considers different species. In genetic diversity, it considers alleles and genotypes of a species, whereas species richness considers various species within area. Part B, Roman number 1, outline the various ways used in maintaining the genetic diversity via breeding programs. Captive breeding will increase population of species due to small gene pool compared to the wild. Inter-zoo exchange of animal for breeding. Zoos select mates of species, preventing inbreeding and avoid genetic drift. Introduce alleles from other populations. Encourage outbreeding. Use of artificial insemination or in vitro fertilization. Measure the genetic diversity via DNA profiling. Part B, Roman number 2. Explain how breeding programs and reintroduction schemes will help increase the population in the wild. Advantageous alleles that adapt to changing are passed to the offspring and the subsequent generations. In this increase in the number of species that are immune to diseases. For example, etc. Use of in vitro fertilization or artificial insemination increases the number of offspring. Reintroduction to the wild increases the allele frequency in the wild population. Part C. Outline how, number 1, a chromosome reaction is triggered in human reproduction. Stimulus. The sperm makes contact with the zona pellucida. A chromosome swells and fuses with the sperm cell membrane. Digestive enzymes like hyaluronidase get released by exocytosis. The enzymes digest zona pellucida and allow the sperm cell to penetrate. Outline how polyspermy is prevented after a chromosome reaction. Sperm cells, they fuse with the egg cell membrane. This triggers the cortical reaction. The cortical granules fuse with the egg cell membrane and released via exocytosis. The content of the cortical granules released into the jelly layer thicken the layer. Describe the transport mechanism involved in the exchange of substances between the mother and the fetus. Part B. Explain how smoking by a pregnant woman may damage the unborn baby. Part C. Describe the similarities and differences between the mammalian male and female gametes. Part A. Describe the transport mechanism involved in the exchange of substances between the mother and the fetus. Oxygen and nutrients will diffuse from the maternal blood to the fetal blood, transported in the umbilical vein, and waste fetal products diffuse from the fetal to the maternal blood from the umbilical artery. The capillaries of the allantois in the villi, they form a huge surface area for the exchange and increase the diffusion rate. Microvilli of villi increase the surface area further. Some exchange by active transport, for example, ions and amino acids. Others by facilitated diffusion, like glucose, pinocytosis, in which numerous mitochondria in the cell of chorionic villi provide energy for active transport, and pinocytosis. Cell surface membrane contain protein carriers, molecules used in the uptake of materials into the villi by active transport. Part B. Explain how smoking by a pregnant woman may damage the unborn baby. carbon monoxide and nicotine either enter the mother's blood across the placenta to the fetal circulation. Increased likelihood of intrauterine growth, restriction, retardation, possibly due to reduction in blood flow through the placenta, and vasoconstrictive effect of nicotine. Carbon monoxide may reduce the oxygen carrying capacity of hemoglobin. Other effects include low birth weight, premature births, respiratory complications like respiratory distresses, syndrome, and birth symphysis. Congenital heart defects like potent foramen ovary and ductus arteriosus or ventricular septal defects, intrauterine fetal death, central venous system defects like cerebral palsy. Part C. Describe the similarities and differences between the mammalian male and female gametes. We shall start with the similarities. Both gametes produced by meiosis. Process of gametogenesis occurs within gonads for both gametes. Gametogenesis for both involves the multiplication, growth, and maturation phases. Both gametes are haploid. Part C. Describe the similarities and differences between the mammalian male and female gametes. Let's look at the differences. Male gametes. Sperms are smaller, about 2.5 micrometer in diameter. Female gametes, the ova, they are larger, about 140 micrometer in diameter. Male gametes cell differentiated into head, middle, and a tail. Female gametes more uniform shape, male less spherical. In male gametes, they are motile. Female gametes are non-motile. Male gametes only produced at puberty. Female gametes, they are present through undeveloped at birth. Male gametes, they are no polar bodies. Female gametes have got polar bodies. Male gametes produced and released in vast number. Female gametes, fewer produced, fewer released. Number 40. What is meant by double fertilization in flowering plants? Part B. Explain how, number 1, sexual reproduction produces offspring identical to the parent. Number 2, sexual reproduction causes variation among offsprings. Part C, describe the formation of a seed following fertilization of the ovule. What is meant by double fertilization in flowering plants? This is where one of the male nuclei fuses with the egg nucleus in the embryo sac to form a diploid zygote. The second male nucleus fuses with the two haploid polar nuclei, or the endosperm nuclei, at the center of the embryo sac to form a triploid endosperm. Part B, explain how, number 1, asexual reproduction produces offsprings identical to the parent. Production of offspring does not involve gametes or sex cells. Offspring thus produced by mitosis or binary fission in amoeba, budding in hydra and yeast, spore formation in muca, fragmentation in spirogyra, and parthenogenesis in male bees. Explain how, number 2, sexual reproduction causes variation among offsprings. Gametes or sex cells are involved. The gametes gets fused. Meiosis is involved in gamete formation, that is, prophase 1, in which the crossing over and random segregation, stroke assortment of homologous pairs of chromosome occurs in metaphase 1. Part C, describe the formation of a seed following fertilization of the ovule. The zygote divides rapidly by mitosis and develops into the embryo, which then differentiates into a young shoot, the plumule, and the young root, the radicle. The seed leaves, the cotyledons, and the primary endosperm nucleus also divides mitotically to give a mass of cells, the endosperm. This remains in grains, while others it is quickly absorbed. Nucellus becomes crushed out of existence to allow the embryo and the endosperm fill the area inside integuments. Integuments become seed coat, that is, the outer integument becomes the tester, and the inner integument becomes the tegumen. Water content is reduced to about 15%. Thank you very much for participating in reproduction in living organisms. Question 36 to 40. Please do like, share, and subscribe.